So welcome everybody to the Bioexcel webinar number 82. Today we will have a webinar on cryo-EM structure refinement using density guide simulation and GROMACS. The presenter of today is Tatiana Shugaeva. She's working at the Royal Institute of Technology, KTH, in Sweden. I'm Alessandra Villa, and together with Otto Andersson and Richard Norman, I'm hosting for Bioexcel this webinar. The webinar will be recorded, and then will be posted on the YouTube channel of Bioexcel. During the webinar, if you want, you can ask questions using the function Q&A that is located at the bottom of the Zoom application. Depending on which operating system, you can see this symbol or this other symbol or another one, but the idea is the same. You can type there your question, and at the end of the webinar, I will unmute you if you have a microphone, or if you don't have a microphone, I will read your question. So please specify if you have or not a micro. That will help speed up us. Thank you. After the webinar, it might be you have still some question about density fit and GROMACS. So then you are most welcome to go to the GROMACS forum at gromacsbioexcel.eu. And there we have created a tag called density fit. So you can post in the user discussion. And if you tag density fit, Tatiana for the next week will be happy to answer to your question if still you have a question. Okay, so now I tell you something on Tatiana. Tatiana is currently is a PhD student in Erik Lindahl's group at the Royal Institute of Technology in Stockholm. She is working in the development of cryo-EM structure refinement methodology using density guides molecular dynamic simulation. But she's also working on molecular mechanism underlying the function of pentameric ligand-gated ion channel. Previously, and exactly between 2020 and 2022, she was working at the Lomonosov Moscow State University with Boris Feniouk group. And she was also working in a pharma company, collaborate with the pharma company BIOCAD, where she was working on improving antibody affinity and model AAV's capsid structure for gene therapy application. And now she will tell us about her last work and density guide simulation in GROMACS.
Yes, great. I hope you see my screen now. Thank you very Alexandra, thank you very much for a nice introduction, and thank you, Otto and Richard, for hosting this webinar. I will be talking today about cryo-EM structure refinement with density-guided simulations in GROMACS. At first, I will give a brief intro in this topic, like what kind of simulations that is, why do we need them. Then I will switch to some a bit more in-depth theory, how do they work, and I will go through the most important density fitting parameters that we can set up in GROMACS. And in the second half of this webinar, I will be talking about the density fitting tutorial that we have released recently with Alessandra. In this tutorial, there is a step-by-step guide how you set up these simulations. This is the QR code that leads to that. So if you want to read the tutorial along with me as I pass through it, you are free to open it. Okay. Let's talk. What is it? What are density-guided simulations? Here we see a short, like a brief scheme, how cryo-EM structure preparation is conducted. And we see that there is a step here called map reconstruction and model building. In this step, we have the density already of the molecule of interest, but we still need to build an atomistic model that corresponds to this density. When doing so, we need to take two things into consideration. The first is obviously atoms should fit well to the density, But it is also very important not to introduce any implausible geometries, any steric clashes, wrong side chain orientations. And if our density has a very high quality, if it's high resolution, then it's fairly simple to do. We even have AI algorithm to do that. But in case if your resolution is low or like parts of the density are poorly resolved, you might want to use an additional source of this chemical information. And MD force field is a very nice source of that. So what if we combined the information from the density and information from the chemical force field in the same simulation? Well, that's exactly what density-guided simulation is. Here we see a simple, slow example, the same one as we will use in the tutorial in the second half of the webinar. And you can see that this helix changed its orientation. When we started, it was vertical, and now it's somewhere a bit more upward. So that was an example. Now, if you really want to use this kind of simulations in your work, here are the materials that you will probably need. Manual, GROMACS manual, user guide, and I really recommend you watching the webinar that Christian Blau gave on Bioexcel some time ago. He's the original developer, and he went through all of the parameters, and he also published a very nice paper in PLOS Computational Biology, where you can find some like practical insights how to use it. I can also, if you don't want to listen up to the end of the webinar or you don't want to go through the tutorial, there's a very quick start guide. Like all you need to try how density fitting in GROMACS works is just a structure file and density file. You can use files from the tutorial. For your structure file, you do standard GROMACS minimization and equilibration. You check that your system after equilibration is roughly aligned with the density. If they're not, maybe it's good to wait up until the end of the webinar where we discuss the alignment. And then you just add 2 lines of code, density guided simulation active and reference density file name, basically that you want to do density fitting and which density do you want to use. And that's all. After that, you can start your MD simulation as usual. But this is a quick guide. If you want to delve a bit more deeply into the theory, let's move on and discuss how does density fitting work. Here we have an example. Blue is the density and red is a helix that is curved, but it seems that we want to straighten it up so that it fits into the density. We need to somehow calculate these forces that will act on atoms and pull this helix inside the density. So how do we do that? It's a bit simpler to explain it in a one-dimensional case. We have a green atom that can only move along the horizontal axis here, and the pink is the reference density. According to the density, it seems that our atom should be somewhere here, like somewhere below the peak. How do we calculate the force that will move it to this part? First thing that we do is that we simulate we made an artificial density around our atom, this green Gaussian curve. Then we calculate the similarity between the simulated density and the reference one. And we can use this calculated gradient of similarity to compute what force should act on our atom to move it to the right. This was 1D case. If we look at it in 3D case, it looks actually very similar. This is our alpha helix, and first the thing that we do is that we generate the artificial density around it that is shown as this red cloud. Based on the similarity gradient between the blue density and the red density, we determine the feed forces. Then we combine feed forces with the normal force field. This is depicted as red arrows, and combine the forces to update the coordinates. Important thing here that we have to balance forces that come from the density and that comes from the force field. Now that you know roughly the idea behind density fitting, we can take a look at the parameters. The list is super long and may look scary, like it's 14 of them, I think. But actually, 2 of them we have already met. The first is to turn fitting on, and then the second is to provide the density file name. The last 2 we also don't really need to think about. They are needed for alignment. We will cover this more in the last part of the webinar. And now we have 10. Among these 10, most of them go into 2 separate groups. As I said, to calculate the forces that comes from the density, we need to simulate the density around our atoms, and this group of parameters controls how simulated density will look like. And the second big group of the parameters is about balancing the force that pulls atoms into the density with your normal force field. Let's move on. Let's discuss the most important parameters one by one. The first one that we can look at is density-guided simulation group. When you do your usual GROMACS MD simulation, you have an index file in which you have atom groups like protein, backbone, side chains. And in this simulation group, you can just write what group do you want to use for a fitting. Do you want to fit the whole protein or maybe just the specific loop or specific residue? So this is roughly straightforward. The second parameter that we should probably look at is the similarity measure. As I said, we need to calculate the similarity between our artificial map and the reference map. There are 3 options. The first is inner product. This similarity metric will give the most strong attractive force to the regions with high density. While the second option, relative entropy, will take overall fit a bit more into account. Like if there will be some regions of your protein that are not fitted, that will affect significantly the similarity measure. And cross-correlation is something in between them. So I would say if you start optimizing your system, start within a product. But if you don't like, if you have some significant conformational changes that you need to occur, then I prefer cross-correlation. Now we move on to the second big group of parameters that describes how do we generate the artificial density around our atoms. The first option here is Gaussian transform spreading width. As you see in this 1D example, the shape of our Gaussian can be different, and with higher width value, we might get a bit more overlap between our artificial density and the reference density. So when picking this parameter, see if you want to model some significant conformational changes, maybe you want at first to set this parameter a bit higher up to allow for this higher overlap with reference density and smooth movement. But for the fine tuning, you can make this a bit smaller for precise atom positioning. Another thing that we should consider for generated density is whether we want atoms to contribute to the simulated density equally, or do we want it proportional to its mass and charge? I think unity is a default one, but I mostly use mass so that hydrogens or some types of atoms could contribute a bit less. And with that said, we can move on to the last big group of parameters, which covers the balance between density force and your normal MD force field. The first parameter that you can consider here is density-guided simulation force constant. It basically defines how strong the pull from the density is. If it's too low, you won't see any movement towards the density. If it's too high, you might see some unfolding, some structure breakage, and even the system can blow up quickly. So this is really the parameter that you should probably tune for your specific problem. Another important parameter in this group is density-guided simulation NST, which defines how frequently do we apply the density fitting force. This one affects your performance significantly. It says 1 by default, which means that we calculate fitting forces on each step, but you're likely not necessarily need this. If you put 2 here, it will calculate fitted forces every second step, or you can put 10 and calculate them every 10th step. If you calculate fitting forces frequently, your density-guided simulation will be like from 2 to 5 times slower than the normal MD1. But if you can apply fitting forces rarely, if you can do it will allow you for a very, very smooth fit, and the performance overhead will also be relatively low. These were like simple parameters, but there is also a very nice additional algorithm that is available for density fitting called adaptive force scaling. The idea behind it is that when we start density-guided simulation, the force from the density is very, very low. And then we see if the similarity between our simulated density and reference density increases. If it increases, it means that everything goes well and we can increase force from the density a bit more. If the similarity does not increase, that means that maybe we need to sample this conformational region a bit more before pushing forward. So we wait and add and increase force from the density only when similarity starts rising. For me, it always sounds a bit counterintuitive, but I actually really like it. I tried it and I think it works pretty well. There are 2 parameters that correspond to that algorithm. The first is called adaptive force scaling. You can set it false or true if you want to use it or not. And the second one is adaptive force scaling time constant, which determines how frequently do we scale the force. And again, the higher it is, the smoother will the force grow. Be aware with this algorithm that since we increase forces as of our simulation, forces will be very high. So it is most likely that your simulation will end up with crash if you use this one. Maybe this is the only GROMACS option with which we can expect crash in the end. But the idea here is that you run it and then you post-process it and pick the frame which you think gives you the optimal fit. And we will discuss this a bit more in the tutorial section. Okay, just a quick note that you can use different density file parameters, like extension doesn't really matter. What matters is the header, and in principle, you can even use the cryo-ET density. So the algorithm is very versatile and works with different file formats. But before starting the simulation, you should really check if the density is aligned with the protein. So don't expect that the protein will automatically rotate or shift to the position that you want it to shift. To check that, you can open the density and the protein VMD or PyMol and look at the results. In the tutorial, we will discuss more how you can align it if they're not. Now I think I still have a couple of minutes for this section, so I can go through some of the recent preprints that came out of our lab with this density-guided simulation algorithm. In the first one, we combined this type of simulation with AlphaFold. We generated an ensemble of different protein conformations and started density-guided simulations to optimize the model building for new conformations of the protein. So check this out if you want to know a bit more about this kind of stuff. And another postdoc from our lab, Nandan Haloy, he recently published a preprint where we can combine AlphaFold 3 with density-guided simulations for ligand position refinement in the binding site. So also check this out for some practical hints. This is the end of the first, theoretical part. Now we can switch to the tutorial walkthrough. These are different ways you can access the tutorial, like the QR code, the link. If you proceed to this link, you will see the page, on this left image. And to look through the tutorial, you can open it as a plain text, or you can use online interactive Jupyter notebook at MyBinder. You can also I didn't highlight it, but there is a Zenodo link in the first paragraph where you can download all the materials. If you want to run this tutorial on MyBinder, then you don't need any additional setup. Just click on the link and the notebook will open. But if you run locally, you will probably need to create a conda environment with all necessary packages. You can use conda-env-create command for that and use environment.oml file that is distributed with the tutorial. Then you need to activate the environment and start Jupyter Notebook. If you do that, you will see something like this. I hope both people who run on MyBinder and locally will see this kind of interface with code cells and some text cells. Here is just the basic information about the tutorial, but if we go further, we will start seeing the code. First of all, I'm sorry that I'm not showing this online, but I think it's simpler to show it as screenshots. And I will show a lot of code and text on some of the slides, but don't worry about reading everything. I will try to highlight the most important thing that you should put attention to. So preparations to run the tutorial. First of all, we will use automatic density alignment tool called rigid body feed. This tool was not available in Conda, so we have to install it now with pip. If you run this cell with an exclamation mark at the beginning, then the rigid body feed package will be installed into the environment where the tutorial is running. So you should see something like successfully installed and rigid body feed after that. Then we want to import the Python packages that we're going to use in the tutorial. NGLView to visualize molecules, MD analysis for analysis, and pandas and matplotlib for data visualization. In some of the cells, you can get warnings, read the warnings, be careful with them, but this particular one is not important. If we move forward, we will see some description of the system that we're using in the tutorial. The protein is called calcitonin receptor-like receptor, and this is a G protein coupled receptor. We also have its cryo-EM density, which I pre-processed. I extracted this part of the density from a bigger complex using Chimera. Here we see that for visualization, we will use NGLView. These are the links to the docs. And if you run NGLView, you will see this nice interactive window with a molecule where you can drag it, rotate it, and use the small arrows in the upper right corner or in the triangle in the lower right corner to change the size of the window. Now, let's move on. Yeah, the density, you can also use NGLView to visualize the density file. As I mentioned before, input structure and density are the most important thing that we need to set up the simulation. If we move further, I have a tutorial pipeline here. For input structure, we will first do minimization, then equilibration. After equilibration, we will align the density to the equilibrated system. And after that, we will be able to start density-guided simulation and analysis. Yes, again, slide with a lot of text, but this is basically standard MD system preparation thing. We use PDB to GMX to prepare topology file and GRO file. We create the box using edit-conf. We use GMX Solvate to add water to the system, and GMX, again, ion to neutralize the charge in the system. In Jupyter Notebook, you can use echo to select the options if GROMACS asks you for certain input. For more detailed explanation of this procedure, you can go to the introductory MD tutorial. This is the slide about equilibration. For certain reasons, we didn't include equilibration, the simulation itself, into the tutorial. One of the reasons was that on my binder, it is relatively slow. So you can start, you can copy the prepared system from the folder with the input. But if you want some challenge and learning, you can set up minimization and equilibration yourself on the basis of other tutorials. This is how the system with water looks like. You can move on. And now we switch to the most important part, probably, maybe not the most, but to the density alignment. If we visualize the density at the same time as the protein, we can see that it looks weird. Like when you will go through the tutorial, you'll be able to rotate it and you will see that it really doesn't look good. So there are two options that you can use for alignment. The manual approach is to use density analysis software such as Chimera X. Here's the link to their official tutorial how to do the alignment. But we will use the automated script called rigid body fit that was written by also Christian, who is the developer of density fitting. This script is relatively easy to use. The only thing you should probably be a bit careful about periodic boundary conditions sometimes. Keep that in mind, but it should be fine with our system. How to use this rigid body fit? We have a equilibrated structure and input density. We use these two files as an input to rigid body fit script, like in this command, rigid body fit, the structure, provide the structure name, density, we provide the density name, and here is the parameter called sampling depth. that defines how exhaustive the alignment would be. It makes the calculation, if you increase this parameter, it will make your calculation a bit slower, but if it's too low, then you might end up with a poor alignment. So run this and check if you like the result or not. As an output, this script gives us a file called transform.json that contains shift vector and rotation matrix. And we don't even need to dive into what does it mean. The important thing that we can just copy this vector and matrix to the MDP file. And GROMACS will know internally how to align density with the protein. To check if we like the alignment result, we can visualize the fitted molecule and the density at the same time. And we see that now almost no parts of the protein are outside of the density, but some are, for example, this particular region. And this is actually where we need some density fitting to move this alpha helix into the new position. This is the cell that describes a simple Python code in which we extract shift and rotation matrix from transform.json and put these values into the correct MDP options, density-guided simulation shift vector and density-guided simulation transformation matrix. And we also provide the density name. And I think that is the whole preparation. We can run GROMPP with the updated MDP file. And then you have, for the tutorial, you have two options. You can start density-guided simulation yourself, but especially on my binder, it can be too slow, so you probably won't be able to run it up until the end in my binder. So if you want to skip this step, I recommend you try running it, see that it doesn't give you an error, that you see that you can start it, but then you can stop the simulation and copy the complete simulation file from the reference folder if you delete this hash sign in the code cell. It's nice that in NGL view you can visualize not only static structures and densities, but you can also visualize trajectories. So we load the trajectory, and you can use these play and stop buttons to control it, and use this slider to switch between frames. And if you look carefully, you can notice that atoms that were previously they were outside of the density, now they are fitted better. Of course, this NGL view visualization is not optimal, so maybe you can additionally use Chimera and other density visualization software for better seeing the result. How do we how can we characterize the differences in the structure in a more accurate way? We can use RMSD calculations. For example, here, this is the plot with structure RMSD compared to the first frame of the simulation. We see that a lot of changes happen in the first, 20 picoseconds of the simulation, and then it is roughly stable. To calculate this thing, we use align trash function from MD analysis package. And we use matplotlib to generate the image that I'm showing. Another thing, since in this tutorial we're using test system, an example for which there is an actual resolved structure that corresponds to our density. And we can compare how different our results, our structure is compared to the actual result structure for this protein. And this is the RMSD plot, and we see that RMSD changes from like 2.2 at the beginning up to 1.4 at the end of our simulation. In this simulation, I also used adaptive force scaling algorithm, and let's look carefully at the video that we see. First, everything is fine, but at some point, we can notice some strange behavior of the atoms. For example, in this region, like at first, helix looked completely normal, but in the end, it has some strange tilts here and here. And these are probably the signs of overfitting, like the force that comes from the density becomes too high and it messes up our chemical information that we had in the normal force field. So how do we pick atoms from the simulation part where everything looks good? The idea, what we can use for that is cross-correlation and GOP score. Cross-correlation is a metric that shows how well the structure corresponds to the density, and we see how it increases over the course of the simulation. And GOP score is a metric that characterizes the geometry of the protein, how like optimal it is or how bad it is. And we see that closer to the end of the simulation, GOP score starts to increase, which demonstrates that some overfitting happens, and probably we want to pick a frame somewhere where cross correlation is not that big, is already big, and GOP score didn't start to increase. For strategies, how to do that, you can check the AlphaFold preprint that I showed before, for example. I think this is all that I wanted to share with you today. Thank you so much for your attention. Thank you so much to my tutorial co-authors, especially Alessandra for guiding, and my whole Stockholm biophysics group that are very nice, and they helped me to learn this algorithm. Thank you.
Thank you very much, Tatiana. Do you hear me now?
Yes, I hear
Okay,
you.
great, because I had some problem with the microphone.
Yeah.
Thank you very much. I was posting in the chat the link to the tutorial if you want to do it. And we have some questions. And the first one is from Mandar. I will see if I can unmute you. Otherwise, I will read the question. Let me see. Where is Mandar? Okay, I allowed you to talk. If you can talk, let me know.
Hello.
Hi, we hear you.
Hi.
Please go ahead.
Yeah, so thanks for the nice talk. Actually, it's interesting. So actually, I'm working on cryo-EM map for like RNA prediction. So is there any range of force constant do you think that like it's ideal for like flexible, considering the RNA flexibility actually?
Oh,
In-
unfortunately, I have never worked with RNA density guided simulations myself, so I cannot comment on force constant, the exact values. But have you tried adaptive force scaling?
No, I don't know. Like this is the first time I'm looking at the tutorial.
Oh, okay, sure.
I can try.
Sure.
And so my structure is also modeled. So let's say if the structure is like far away from the map, then that will be a like issue, right? Like it will be a
Yes. You need to ensure that the map overlaps with the structure. You will
so
probably need to run the alignment.
Is there any parameter like you consider as like, okay, between the map and the predicted structure that like we can think
Uh, can you
with?
repeat, please? What do you
So
mean?
let's say after aligning the map and RNA structure, I'm
Yeah.
not sure whether RMSD is the parameter here, but is there any way to like quantitatively say, okay, like we can use this approach in certain case or like not, something like that?
Do you mean if the alignment is good?
Yeah, if the alignment, like how much like percentage alignment, let's say, should be there or something like that
Um, I
in
mean,
your experience.
even if the percentage is low, I guess in Chimera you can you can use fit and map function. You can find it in, for example, in the tutorial that I showed you, like showed the link in the slides,
Yeah.
and. Uh, I will always worked with values of this correlation around like 0.6, 0.7. But if you do a very mild, slow fitting, even the poorly aligned protein can actually, or your RNA complex can shift towards the density. So just try, if the overlap is poor, try to try use adaptive sampling and set the initial force constant very low, like
Okay.
maybe 10 E1, something like that, and look at the results.
Okay. Thanks. Thanks. That's a great suggestion. Thanks. I'll check. Yeah.
Thank you so much for your question. Oh, Alessandra, I don't hear you.
Thank you very much, Tatiana. Now we go further, sorry. Now we have an anonymous attendee that so has a couple of questions since he does not have a microphone or she does not have a microphone. So the first question is, what is the difference between density of atoms and collection of atoms?
Collection of atoms. Sorry, I'm not sure what does that refers to. But maybe what
Maybe
is
it means a group of atoms. I don't know if you have used the word collection, but yeah.
yes, maybe a group of atoms. Then during the algorithm, we generate the density around your protein system, which might look kind of similar to the cryo-EM density that you can download from the EMDB, while the protein group of atoms is the just atom numbers that you want the force to be applied to. So, for example, if you want to do a fitting of your protein for the parameter group, you can print protein, which will mean that you want this set of atoms to be fitted.
Okay, thank you. Then I will skip the second question I read, but I don't also myself understand completely. Why opt for density guide simulation over classical MD simulation? That I think is, I don't
Well,
understand.
I mean, it depends on your purpose. If you want to fit into the density, you will probably want to add some fitting force.
Yeah, the force is just being just used to guide atoms toward the protein. Does it stabilize the protein? Is this it is in any way related to applicable physiological pH?
Um, First of all, it is likely that the conformation that you have in your density is something physiologically relevant and stable. So if you shift your atoms toward this conformation, I would expect it to be more or less stable. And also regarding pH, when you do your simulation setup, you can pick different protonations for your atoms, depending on what pH do you want to use. And if you also want to know a bit more about this, we in GROMACS, we have also recently added the constant pH algorithm that you can also look into if you want some more fine tuning for pH in your system.
Thank you. And we go further now. We have a question that compare the methods with the methods. I ask a comparison and the difference between what is implemented in GROMACS and with an approach that is called ISOLDE that is implemented in Climera X. So I don't know if you're familiar with this other approach. Anyway, the question is how much different is the density guide simulation in GROMACS and in Climera X ISOLDE? And does it in principle different from using EM map as structural restraints?
That's a good question. And I think I am not super familiar with ISOLDE. I didn't use it that much. But I think density guided simulation might be more useful in the more complicated cases because you have a lot of parameters to fine tune, and you can try to simulate maybe more significant conformational transitions compared to ISOLDE. And I guess it is different from just applying density as a restraint because the algorithm is more versatile and more tunable.
Okay, thank you. And then we have another question from Andre Graca. So the question here is periodic boundary condition. Could you explain what do you mean by that? Could you remind the exact name of the rigid body fit tools package developed by Christian Blau?
Sure. Regarding the periodic boundary conditions, this is actually very good question. I didn't mention it in the webinar itself, but this is something you should keep in mind, that we do not apply periodic boundary conditions to the density. We don't have copies of the density, and sometimes your density cell can be even bigger than your simulation system. Like in this slide, the blue box is the density box, and the smaller ones are the simulation boxes. And the algorithm is written the way that density fitting only takes into account the instances of the protein that are closest to the center of the density. So, for example, here we have two periodic versions of this polymer part, but density force will only apply to this copy. So that's what I meant, that you should be careful with that. And regarding the name of the tool, it is called rigid body fit. For some reason, I also struggle to find it on Google, but we will upload slides on Zenodo, right, Alessandra?
Yes.
So
Yes.
all the links, you'll be able to use them from the slides.
Yes. Thank you very much. And then we have a question from Anthony, and I will allow you to talk, Anthony, if you can. No, I think
Yes.
Anthony
Do you hear me?
cannot. Oh, yes, we can hear you. Please go ahead.
Okay, thank you, Tatiana, for your talk. I have two short questions. Is there any impacts of the size of the polypeptide chain? That's the first question. And the second question is in the same way, is there an influence between simulation of one polypeptide chain and complex protein, DNA, or protein ligand? Is there any impact of the size of the system in some way?
Well, the kind of obvious answer that size will strongly impact the performance that you will get. Because density fitting is also super slow for big densities and big complexes. Also, you might you might check if you need other force constant values for bigger systems. And another thing, I haven't worked with DNA protein complexes myself, but I remember that there might be some issues with different attractive forces from DNA residues and protein residues. So you might want to try and experiment with the option that I showed here, where atoms correspond with they correspond to the simulated density proportional to their mass or proportional to their charge. So I would say if you see some results are not good, maybe you want to change this parameter.
Okay, thank you.
Thank you.
Thank you very much. So then we go further we have a lot of questions, so we have to be a little quick if we can cover. I think there is another question about, but I think you already answered about multiple chain fit and what is the best approach to deal with the protein-protein complex or oligomers, but I think you already answered. Is correct?
Well, in principle, it doesn't change that much. The setup is the same.
Yeah. So then Mandara has another question. He asks if it could be possible to combine with replica exchange.
I haven't tried that myself, but I think it should be.
And then if yes, will replica exchange use MD energy plus penalty from cryo-EM restraint to decide the exchange probability? Not sure.
Sorry, I'm not sure. Since I haven't worked with that, I'll prefer to. I might misguide you.
I'm not so sure. I think exchange, yeah, but has to be checked. Yeah.
I think I can double check this with Berk later, and if you post this question on the forum, I may come back later and try to help you with that.
Okay, great. Thank you, Tatiana. And then we have there is a question that about MD-based method with Rosetta, Monte Carlo based, and he's asking if you never compare MD-based method with Rosetta, Monte Carlo based method, I think for density fit in terms of performance and quality of the model.
No, sorry, I haven't.
Then we have Victor raised that Victor does not have a microphone, so I will read it. He asking why not simulating vacuum?
That's a nice idea. When I started working with density-guided simulations, I was trying to simulate my systems in vacuum, but it is very fast and it can even work in certain cases. But what I noticed that when you want a significant conformational change, the charges that are not shielded with the solution around the protein they have a very strong impact. So it is. I know that simulating in water is slower, but it you will get much more physiologically relevant result. However, maybe it's a good time to mention that that in the tutorial we're simulating G protein coupled receptor that is in principle a membrane protein. But we noticed that since the force from the density is so strong, you can actually do density fitting for membrane proteins, but without membrane in your system. It was shown in one of the Christian's paper, Christian Blau papers.
Okay, thank you. And then I have a follow-up question. How to deal with noise and resolution when generating the density used for density similarity?
How to deal with an
noise and resolution?
Well, for when we generate artificial density during this during the GROMACS run, of course, it is much less noisy than the reference density that we want to fit in. And if the question is about the quality of the reference density, if there is some noise in the reference density, then I might say that in my experience, of course, it's good to get rid of the noise manually if you can do that in Chimera or somewhere else. But in some cases, it doesn't affect the result so much because anyway, you will have the highest density in the parts actually corresponding to your system, most likely.
Thank you, Tatiana. Now I just jump around with a question because we are almost done. So I think I take the last question.
Yes.
There is an anonymous attendee that has asked, may I ask how you compute the artificial density of unaligned atoms before the similarity gradient evaluation? Sorry, I'm not sure that you cover this.
Can you repeat the beginning of the question? Sorry.
The question: how to compute the artificial density of the unaligned atoms before the similarity gradient evaluation?
Well, first of all, if you want to run density fitting, this generation happens automatically inside GROMACS, so you don't need to do anything yourself. But in case you want to generate artificial density around your atoms, there is a Chimera command for that, maybe molmap, if I remember correctly, but you can look this up and you can generate densities in Chimera, for example.
Yeah. Thank you very much, Tatiana. I think our time is over. I will all the questions that I had no time. We had no time to address. Please post it in the forum and in the GROMACS Bioexcel forum. So gromacs.bioexcel.eu, and tag it with density fit. So Tatiana in next week could answer. Thank you very much. And if you stop sharing, I just want to make the announcement of the following webinar. Thank you very much, Tatiana. We will have we will have next week Elisa Fada with a webinar on restoring protein glycosylation with GlycoShape. And then the following week, so the 18th of March, we have Siverian Margik perspective on simulating whole cell with Martini. I'm looking forward to see you also on this webinar, and I thank you, all of you, for the participation, active participation, the question, and Tatiana for the nice presentation. Thank you, everybody, and see you next week. Bye-bye.